Welcome to our introductory lesson on cell division. So as you know, all living things need to have a way to be able to reproduce to pass their genes on to the next generation. Some organisms reproduce asexually, where there's only one parent involved, and other organisms reproduce sexually, where you have two parents and the offspring is a blend of the two parents. Here we see two paramecia. They are just in the process of reproducing, and the two paramecia here are clones. They're exact copies of each other. They have reproduced asexually. You see a human family, and of course humans reproduce sexually, which leads to very diverse traits. So you can see that this family, none of the organisms are clones of each other. They're each unique and they have their own genetic information. So how do cells divide? Before we get to these multicellular organisms like humans who are very complicated, let's get down to the basics. How do cells actually divide? And it turns out that prokaryotes and eukaryotes divide differently. So if you remember, prokaryotes are small, simple, and they lack a nucleus. They include bacteria. And they reproduce through something known as binary fission, which we'll take a look at next. Eukaryotes are large, they're complicated, they have a nucleus, they have membrane-bound organelles like the mitochondria, and they reproduce through something known as mitotic cell division or mitosis. On the top, you see binary fission occurring, and down here you see eukaryotic cells reproducing. Before we talk about prokaryote reproduction, I want to discuss what DNA, genes, and chromosomes are. So DNA is made up of two long twisted strands, and it contains all the genetic information. This is what DNA looks like down here, right? It's a double helix. It's called a twisted ladder. You've probably heard about DNA before. Now, out of this DNA, there are segments called genes, and genes are basically the parts of the DNA that code for specific proteins and cause specific traits to happen. So for example, you have a gene that helps you produce insulin, which helps you control your blood sugar. And you have genes that create the protein melanin, which causes your skin to have a certain color. So genes are the parts of DNA that actually make traits happen in individuals, and they get passed on from parents to children. Now, genes are organized in units called chromosomes, and you see that this is all this DNA, including the genes, and all of it gets wrapped up into these chromosomes. So now let's look at prokaryotes. Prokaryotes, it turns out, have only one chromosome. They have that one singular loop of DNA. They don't have a true nucleus, just this one loop of DNA, which is quite long. And all of the genes for the prokaryote are on this one loop of DNA. All right, so that's what the chromosome is like. And since it's so simple, prokaryote reproduction is quite simple as well. So binary fission actually means dividing in half, and that's essentially what a bacteria is going to do. You end up with two identical cells, and the steps in the process are quite simple. So let me show you that in pictures. So here we have our original parent cell, right? We have our bacteria, and it's getting ready to divide. So the first thing it's going to do is it's going to copy its single chromosome, so now it has two copies. There's one copy, and there is another copy. The next thing it's going to do is that this bacteria is going to elongate, and the copies are going to go into two separate sides of this long cell. And then what happens is basically the bacteria is going to pinch into two new cells, each getting a complete copy of the DNA. This is called binary fission, and it is quite simple. Here you can see an electron micrograph of a dividing bacterium. You can see the DNA on the two sides and the two daughter cells. So let's take a look at how this actually happens in real time. So what you see here is E. coli bacteria reproducing. You can see the binary fission taking place, the bacteria elongating and splitting into two, elongating and splitting into two. So bacteria can reproduce quite quickly, not this quickly. This is, of course, sped up, but once every 20 minutes, right? So you can build up a huge amount of bacteria in a very short time because they're simple and they reproduce quite quickly. Eukaryotes, on the other hand, if you remember, are more complicated. They have a nucleus. It's going to take more steps to reproduce a eukaryotic cell. So eukaryotic DNA is not in a single loop, in a single chromosome. It's actually divided into segments, into multiple chromosomes. In fact, we humans have 46 chromosomes. Now, eukaryotic chromosomes are composed of something known as chromatin, and chromatin is the DNA and some proteins. So this is what it looks like. You have your DNA, right, that twisted ladder, and it's going to coil around these proteins called histones. Histones are a type of protein. So the DNA is coiling around these proteins, kind of like yarn would coil around a spool. And this structure, the DNA wrapped around the protein, is called a nucleosome. So that is the structure of chromatin in a eukaryote. Now, when a eukaryote is getting ready to divide, it needs things to be way more organized. So usually the DNA is loose and kind of tangled, and the individual chromosomes are not visible. When the cell is getting ready to divide, the chromosomes, first of all, they get copied just like they did in the bacteria. But not only do they get copied, they condense and they form that characteristic rod shape, right? So here you can see chromatin where the DNA is kind of loose. It's in the form of nucleosomes. It's wrapped around those proteins, but you can't see the individual chromosomes. And then what happens when the cell's getting ready to divide is those chromosomes condense and you start to actually see that chromosome, that rod-like shape. Each chromosome is made up of something known as sister chromatids that are connected at a centromere. So what are these things? Well, the sister chromatids are exact copies of each other, because if we want to make two new cells, each cell is going to need to have its own copy of the information. So this side is one sister chromatid, and it's an exact copy of that side, which is the other sister chromatid. And the two copies are held together in the middle by something known as a centromere. So this is what the structure looks like. You have the DNA, just like we said, coiled around these proteins called histones, and that forms a nucleosome. And that gets coiled further and further and further and further until you end up with these chromosome structures, these rod-like structures that are useful in cell division. So this is how DNA looks right before the cell's getting ready to divide. And here are some better views of that. Here's an individual chromosome, right? And a chromosome, of course, like this, is made up of two sister chromatids. Again, two copies that are going to get separated during cell division, held together in the centromere. So this is what we mean, right? This is a normal chromosome. When we want to divide the cell, we make a copy of it. We hold the copies together. And then when cell division happens, one copy is going to go into one daughter cell, and the other copy is going to go into the other daughter cell. So let's pause to talk about humans. So humans have their DNA in 46 pieces and 46 separate chromosomes. It turns out that humans are diploid, which means they have two sets. So they have 23 from one parent and 23 from the other parent. So 23 from mom and 23 from dad, reaching a total of 46 chromosomes. Now, all these chromosomes are in pairs, right? One from mom, one from dad. And here we have a picture called the karyotype that shows you a human picture of the human's chromosomes. So you have one from mom, one from dad, one from mom, one from dad, one from mom, one from dad, until you get to 23 pairs. Now, all of these are in homologous pairs. That means that these chromosomes come in pairs that are the same length, they're the same shape, and they have similar genes. Like they might have a gene for eye color, and then that means this one also has a gene for eye color. So they have similar genes on them. And all of these chromosomes are in homologous pairs, with the exception of one. And that's in males. In males, all of them are in pairs except for the last two, the sex chromosomes, because males have an X chromosome and a Y chromosome. And you can see right here that they are not the same size and they are not the same shape. And I'm going to tell you that they don't have the same exact types of genes. So those are not homologous. This karyotype is for male. You can see it close here. So a karyotype is an organized picture of chromosomes, and you can see them all paired up into homologous pairs, and then you see the sex chromosomes right here. What does the karyotype of a female look like? This is what it looks like. Again, you have all these chromosomes having nothing to do with sex, they're autosomes. And then down here, you see two X chromosomes. That's how I know that this is a female. Okay, so how do eukaryotes reproduce? They reproduce through mitotic cell division, and this is an asexual process. Mitosis is used to grow a fertilized egg, a zygote, into an adult. Mitosis is used to grow in size in a multicellular organism, because if you remember, organisms grow not by the cells getting bigger, but by producing more cells, right? And we said how the whale and the tiny little mouse have the same size cells. The whale just has more. And then mitosis is also used every time you have dead and worn out cells. So your skin cells are constantly dying and being replaced. Your red blood cells are dying and being replaced. Cells inside of your stomach are dying and being replaced. And that happens through mitotic cell division. And then mitotic cell division is also how single-celled eukaryotes reproduce so that they can pass on their genes. This is the eukaryotic cell cycle, which we're going to take a look at next, but we'll just mention them briefly here. So there's an interphase part of the cell's life, which is divided into G1, S, and G2, and we'll look at what those are. But basically, an interphase, a cell is going to grow, copy its DNA, and get ready to divide. And then you have this phase over here, the M phase, which is mitosis, which is how you're dividing the nucleus of a cell. And then you have cytokinesis, which is how you divide the cytoplasm in a cell. And we'll take a look at all of this so it makes more sense in the next lesson. But I just wanted to give you an overview of how this fits into the human life cycle. So let's start here, right? We have an egg cell and we have a sperm cell, and they come together through the process of fertilization to form a fertilized egg known as a zygote. And that's really the first cell that you ever wore, right? This was your very, very, very first cell, even before your parents knew about you. And then this one cell went through mitotic cell division, right? And it made copies and copies and copies. And this part is asexual. You have this zygote making copies to form a ball of cells and forming more and more and more cells. And we've already discussed differentiation, but this is where differentiation happens. You have this ball of cells, and then each of those cells is going to differentiate. It's going to become specialized. Some are going to become nerve cells. Some are going to become skin cells. Some are going to become the cells that make up your internal organs. Some will become blood cells. So differentiation occurs until you form a human being, right? A female, and we have a male. And then to continue the life cycle, females have ovaries in which they produce eggs, and men have testes in which they form sperm cells. And this process here is called meiosis, which we didn't learn about yet. We'll learn about in the future. But during meiosis, sperm and egg are formed, and then the cycle can repeat. So I hope that was a useful introduction to mitotic cell division and how prokaryotes and eukaryotes divide. And next time, we're going to take a look at this stuff. This is mitosis. So thank you so much for watching.